And then I'll go ahead and I will turn it over to the group for any questions.
I guess, so for the format of the tests, what kind of questions is it going to be? Multiple choice, true, false? What type of questions are going to be on the test?
That's a fair question. So it's kind of split into two parts. The first part is going to be a mix of true, false, multiple choice. And then the second half of the exam consists of about 4 to 7 short answer questions, depending on how many points they're worth. So they're worth 100 points total. So 50 points will be a series of, like I said, the multiple choice and true false questions. And then the second 50 points will be in the form of short answers that range from about 4 points to maybe 15 points per question.
Awesome. And then I know there are some equations that are in the cog books. Are we getting those equations, or do we have to have those equations memorized?
So it's probably like the species diversity one. They're not on the exam themselves, so you'll have to know them. There's only a couple equations that you'll have to know for this exam.
Okay, awesome.
So we have one message in the chat saying they'd love to go over the learning objectives for exam 1. We can do that as a group, unless anybody has any specific questions outside of those questions they wanted answered first.
I wanted to know, did we need to focus so much on definitions, or more so like the bolded definitions in cog books, or more so like define evenness and use that, and like ecological envelope?
You will have to know what the definitions are. I mean, you're not gonna get a question that says define ecological envelope. It might say describe how an ecological envelope can be used to measure species ranges.
Yeah.
It would be more conceptual based and more like thinking questions, but you will have to know the base definition to be able to answer that type of question.
Okay, so it's less about like what S observed represents, like, do you know what I mean?
Yeah, so in that equation, species of, it's not going to say what does S observed mean.
Perfect. Okay, cool.
Here's a table of data, calculate for community A and B.
Perfect.
Estimated number of species.
Okay, awesome. Thank you.
And it might ask a conceptual question of give you a scenario and say, why would you estimate community A to have less species than community B?
Okay.
Are there any other more specific questions or course related questions? If not, we can then just go through the topics and learning objectives. But the way I want to do that is I want to do it as a group and kind of walk the group through it and have somebody, if they can, have you guys answer the questions more than me just read, the answers to you. So I'm going to ask for your participation to do this. So the first question is contrast 3 ways that biologists define biodiversity. So if somebody just want to list off the 3 types of diversity that cogbooks talk about.
Ecosystem, species, and genetics.
Yeah, so those are the 3. And then do we want to define those 3? So what is ecosystem diversity consist of?
Varying species within a community.
Yeah, but what about that community, I guess?
Different habitats available.
Yeah, exactly. So there'll be different habitats available. So this ecosystem itself can be diverse. And what can be different about that habitat? What's an example of different habitats?
Humidity, elevation, different prey or predators in certain areas.
Yeah, so physical
competition.
Yep, no, those are great examples for sure. They can be a little more general too. You could have, you know, a wetland next to a forest. That would be, if that's in the same region, that's a little bit, the diversity there would be 2. But there's even more diversity if you want to get more specific, as mentioned, that it could be differences in temperature, differences in cover, different types of plants within that forest ecosystem itself. So that all contributes to the ecosystem diversity. Now, what about species diversity? What is that when we say species diversity? What do we mean?
There could
Just
be,
the,
Oh.
sorry.
there you go.
I would just say the number of different species that inhabit the ecosystem.
Yeah, so that one's a little more specific, right? So that's the number of species in a given area, exactly.
Yeah.
Now, what are we talking about when we say genetic diversity?
The opposite of inbreeding. So
What do we get when we do
many
that?
different alleles within a species keeps this species healthier. It also helps prevent extinction. It's easier for them to adapt to changing environments.
Yeah, so I think somebody in the chat mentioned heterozygosity. We're talking about the loci, allelic variations. We're talking about alleles. So just different genetic diversity, having more than one type of allele within that environment, and it's healthy for that the species and environment, right? Because we mentioned inbreeding in the beginning. Why is inbreeding dangerous, like for a species survival?
It causes genetic mutations.
Yeah, it could lead to some harmful mutations if we do some inbreeding, and also just not having that allelic variation or variation of alleles. If a disease comes through and their immune system is very, you know, homogeneous, they're all likely going to be susceptible to it and die, right? So if you have a couple different immune responses based on your alleles, some might die and some might survive.
Kind of like the problem with the Cavendish bananas.
You want to go into detail a little bit about that, or briefly, I guess?
Well, the bananas that we know as bananas are not what used to exist. And so I don't remember exactly what it was, if it was like what kind of disease it was that wiped out all the bananas that used to be. So anyway, the only way for us to make more bananas is to go off this one species.
Yeah, and that was
sorry, it's limited genetic. There's no genetic diversity in the bananas that we eat. They're all basically clones.
Yeah. More familiar with that now that you mentioned it. And part of that was selective breeding, right? Where they selected for increased size and growth speed, but sacrificed like not their immune system, but their ability to fight off disease a little bit. So it's kind of trade-offs when you do that type of breeding.
Yeah.
I think another example that was given in like one of the extra videos on cogbooks was the potato famine, where I mean there was a lot of factors that went into it, but because Ireland had only like one kind of potato, you know, this plague came in or whatever and just annihilated all, you know, eradicated all the different, like that one species of potato that helped cause that potato famine.
Yeah, because they have very limited genetic diversity. They're susceptible to disease. So yeah, I think that covers number 1 pretty well, unless anybody has anything they want to add. All right. Sorry, go ahead.
Sorry, I missed like the first couple minutes of being in here. We said that would the equations be provided to solve like Simpson's index or like the community sampling?
Generally, they're not provided. I can take, well, okay, I'm taking a look at the exam here, and you are going to have to be able to calculate Simpson's index diversity, and it does provide the equation on the exam for Simpson's.
Okay, just not for the other one. I can't remember what it's called right now.
Yeah, estimating species
Yes, for estimating species. That one's not provided.
No, that one is not. You will have to do a calculation on that. And then there will be some conceptual questions on there too.
Okay, okay.
Okay. Then number 2 and number 3 kind of go together, I would say. The number 2 is what we were kind of just talking about, estimating the number of species in a community. So does somebody remember what that equation is? Want to share it with us? So it's S equals something.
Species observed.
Yep.
Species observed plus it's like the number of individuals number of species represented by one individual
Mhm.
with the parentheses, that number -1 over 2 times the species represented by 2 individuals plus 1.
Yeah, and everybody's clear F1 and F2, what those mean in the equation. Right, as was just said, it's F1 is the number of species represented by one individual, and F2 is the number represented by 2 individuals. And sometimes that can be 0. Like one of the examples in Cogbooks, it had community A and B, I believe, or communities A and C maybe had.
It was 5.
It observed at least one individual for every species, but then community B had some species that had no observations. So that you would put a 0 in for either the F2 or the F1.
That one confused me. And that's why, like, when I typed in Cogbooks, I was like, I think I'm doing this wrong. But then I figured out it was the 0.
Okay. And then Simpson's index is similar. It's just a different way that scientists use to compare diversity and evenness. And that equation, did anybody have any problems with it, or do they understand what the content of that equation is? Right, it's 1 over the sum of pi squared, and you'll be given a table with those columns and those values, just like the activities, and you just kind of have to be able to remember how to plug it into that equation. And the pi is just the relative abundance values that you're given. Does anybody remember what the range of the Simpsons index can be? Like, what is the minimum value you should expect?
1.
Yep. And then
The max.
go ahead.
I was just gonna say the max would be equal to the number of species within the community.
And how do you, how would you get the max value? When would that occur? What would the scenario of that, or what would that community look like? Referring to, I guess, abundances of the species. Oh, in the chat, yeah, somebody said when they have equal abundances. Yeah, so you only get that maximum value when all the species in the community have equal abundances. That makes sense? So if they all have, say, you have 4 species and they all have 5 observed, then you could get the max value. But if you had 5 observed for one species, 4 for another, 2 for another, then you can't get the max value for the Simpsons index. So it'd be good to know, like, what would be required for the community to either get a value of 1 or the max value. You might get asked a conceptual question on that. All right, I think we can move on to 4 then. Go ahead and stop me. Yeah, anybody, if I'm moving too fast or you have more questions or comments.
What would cause the Simpsons index to be a minimum value of 1 then?
So the equation is 1 over the sum of the relative abundances. So you'd have to have a value of 1 in the denominator. How would you get a value of 1 in the denominator?
If there's only one species.
Yeah.
Okay.
Or there's only one species observed. Yeah. All right, then number 4 asks, says define evenness and use rank abundance curves to compare the evenness of species between communities. So let's just start with defining evenness. Yeah, so in the chat, we got an answer of species evenness is the comparison of the relative abundance of each species in a community.
So, for that one, I kind of like broke it down as equal abundance equals greater evenness, and lowest evenness is there's one species that's abundant and then the rest are not.
Yeah,
Is that okay? Okay, okay.
I follow that. I think it might help. I'll share my screen. And these are the curves, like the rank abundance curves, just so we're all understand what we're, remember what we're talking about. So let's compare communities B and A in this first one on the left. Which community has more, greater evenness?
Community B.
And why is that?
It looks like it's one individual species.
No, these dots each represent a different species, but what is on the y-axis?
Oh, okay. It's more abundant in community B.
Well, if we're talking about evenness, is the abundance different in community B across species? Or are they the same?
They're the same.
Yeah, right. It's very even community because
Mhm.
all the species have the same abundance versus A, you have different abundances for the different species. So the evenness is less for community A than it is for community B.
Okay.
But what about the species richness? Which one has greater richness, A or B?
A.
What is your reasoning for that?
Oh, no. B, because there's consistent varying species within the community.
Let's back up a step. What is richness?
Richness is going to be various species, right?
The number of species in a community, right? Yeah, so somebody else has a different answer in the chat. They say it's the
Oh.
same. What is the reasoning for this potentially being the same here?
Both community A and community B each have 5 different species.
Yeah, exactly, right? So there's 5 dots. So that's the number of species. So the richness is the same because they have the same number of species.
Oh, okay.
What about if we look at B now? Is there a difference in richness in this community?
Yes.
What's the difference?
That B has less than community A.
Yeah, right. There's only 5 species compared to, I don't know what that is. There 16 species, I think, right?
Uh-huh.
Community B. So it has greater richness. But which one has greater evenness in community B?
The one table A.
What do you mean by table A?
You said which one in community B has greater evenness?
Yeah, the eucalyptus or the primary forest.
Oh, the primary forest.
And why does it have greater evenness there?
Because there's like a consistent amount of species in the primary forest.
Yeah, so the relative abundance is in a narrower range, right? So it's in a smaller window compared to the primary forest is wider.
Mhm.
So these are types of questions you might get asked, like which community has greater richness or evenness and why?
Okay.
So the eucalyptus plantation has the greatest evenness.
Yes,
And
because it has it's over a narrower range of relative abundance.
okay.
And like this one over here, right, that had the greatest evenness because the relative abundance was the same for each one. So it was extremely even, as even as you essentially can get. All right, any more questions about defining evenness and using rank abundance curves to compare evenness and species richness? All right, we move on to number 5 then. So this one says contrast alpha diversity, beta diversity, and gamma diversity, and then explain how each relates to the other. So let's just start with defining the 3 terms. So what is alpha diversity?
It's looking at the number of species in a small area like a stream.
Yeah, so in a small homogeneous habitat, exactly like a stream is the example. Is there another name for alpha diversity?
Local diversity.
Yeah, so that's, you can think it locally, geographic, small area. Yep, in the chat too. Good job. What about gamma diversity? What is that? So another name from the chat, somebody said it's also called regional diversity. And when we think of local versus regional, how would we describe it in terms of area? So someone said like 27 streams, so a larger area. Yeah, so again, it's the number of species in all the habitats that comprise a large geographic area. So an example could be a city maybe. Do you typically find more species at a local scale or a regional scale? Somebody said regional. Why is that? A larger area, more space, more resources. Yeah, I think those are good answers. So if you have more space and more resources, what do you have less of between species? Yeah, exactly, competition. Okay. And then what about beta diversity? What is that? In the chat we have, there's the number of species that differ in occurrence between 2 habitats. Exactly. So if you have 2 different streams, or a stream in a pond, or a stream in a field, maybe it can be the number of species that you differ when you sample those 2 different environments. Any other questions about alpha diversity, beta diversity, and gamma diversity? We can move on to number 6. This says, describe how the rate of colonization and the rate of extinction combine to determine the number of species on a region such as an island. So what are the factors that we're going to consider for rates of colonization and rates of extinction that relate to an island? What do animals have to do to colonize an island? So someone said the amount of resources. That is definitely part of it. The less resources equal higher extinction rate. What makes for more or less resources on an island? What about the island itself? Why would one island maybe have more resources than another island?
The size, so larger islands will have more resources than a smaller one.
Yeah, okay, in the chat it's saying size of the island. Another one's how far from the mainland it is, how habitable it is. Larger resources has more, larger islands have more resources. Yeah, so this is speaking in generalities, right? I mean, you can have some instances where small islands maybe have more resources, but in general, all things being equal, a larger island will have more resources because it has more space, and therefore it leads to lower competition. And if you have a larger island with more resources, less competition, then you have higher or lower rates of extinction. Yeah, lower, right, from the chat. Because you won't have to compete as much and there's more resources available. So what about when we talk about distance from mainland, when we go near and far? If you're far from the mainland in island 2 compared to maybe say island 1, what is the rate of colonization going to be, higher or lower for a far island? Yeah, from the chat, lower. And why would it be lower colonization for a far island? One answer is not easy to access, further for animals to travel. Yeah, so you might be limited. Insects might not be able to fly far enough to go to a far out island, but they can go to one that's closer to the mainland. So you have to have more, stronger flight or be able to swim or have, the ability to travel and get to that far island is more difficult. And then I'll share my screen again with this, because now we're going to get the questions asking to determine the number of species when this is taken together. So what are these dots right here at these intersections when we talk about near and far and large and small?
It's the amount of species that would be present on that island at any given time.
Yeah, and the word for it from the chat, somebody said equilibrium, right? Yeah, so these, based on how far it is and how large it is, you're going to have different points of equilibrium for rates of colonization, rates of extinction, right? So if it's small and far, the equilibrium point's over here, versus if it's large and near, the equilibrium point will be over here. Does that make sense? And the equilibrium point is just where the two lines intersect, right? So this is for the small and the far, they intersect right here. So it's just figuring out where they intersect will be your equilibrium points. Are there any other questions about rates of colonization and extinction for this question? Okay, if not, we can move on to number 7. So this kind of builds off of question 6. So this says, use the theory of island biogeography to explain why larger islands usually have more species. So this is just essentially a specific, and we covered it, but let's just make sure we're on the same page and you can do it again. So in this specific example, why larger islands usually have more species. Yeah, so this gets back to what we said before, right? Larger islands usually have more resources. And with more resources, it can reduce competition because you have increased space, so they're spread out a little bit more. Yeah, exactly. You guys are getting the hang of this pretty well. All right, so let's move on to number 8 then. So this one is about fragmentation. So it says, explain how the fragmentation of habitat can enhance or inhibit biodiversity. So why don't we take it piece by piece? So the first one, how can fragmentation of a habitat enhance biodiversity? So from the chat, we have an answer. Different sectors of the island can have different species living on it. What do you mean by that a little bit? So if we have the island, let's call it a sphere, and we have two different regions of it. We have species that are separate. How does that enhance biodiversity? But sorry, to clarify, we're discussing enhancing. From the chat, there's a question. So if we talk about different sectors of the island having different species, okay. Okay, so that's such as like different portions of the island can be inhabited by different species, created a more diverse area. So what, with fragmentation, it could create maybe different habitats, right? And if you have different requirements of different species, I think this is where this person is going with this, then you can have, they're not competing maybe in the same area, or it just creates different types of habitats by fragmenting it so that you can meet the needs of different species and then increase or enhance the biodiversity of that area. Another chat answer, fragmented forests may have different abiotic factors beneficial to certain species near the edge, creating more diversity. Okay, so that's a good thing. If you fragment a forest, you create more edge. So you might get an increase in edge species coming to that area. So on the flip side then, how could fragmentation inhibit biodiversity? I think there's a answer alluding to this previously in the chat saying the total amount of habitat decreases when you fragment it. So that's good. And then another answer, crossing the patches would be risky, limiting migration. That's possible too. So if you have animals that typically live in the interior for protection or cover, and they have to cross, so maybe you have two patches of forest and a field in between now, they're much more exposed and visible to predators. So it could be risky for them moving. So somebody said more competition, and that could be related to less habitat available means less resources, which generally increases competition. So that's good answers. And if you have smaller areas, you generally have smaller populations. And do small populations generally have an increase or decrease risk of extinction? Yeah, right. So the smaller the population, they have an increased risk. So by shrinking the area, shrinks the population and increases their extinction risk. Awesome. I think that covers that one pretty well, how it can either inhibit or enhance biodiversity. So unless there's any other questions or comments, I think we can move on to number 9. So this says, use the concepts of fragmentation and connectivity to compare different designs for a nature preserve. So you had an activity, like I think it was a quiz activity in Cogbooks, where it talked about, it gave you a bunch of different examples. So what are some features about fragmentation and connectivity that relate to biodiversity? How would you create a nature preserve if you had to talk about it? So here we go.
So you could have, instead of like having 4 little reserves, you could have 1 large one. Or instead of having like, you know, all these reserves lined up in a row, you could have them more clustered together. And, or if they are in a row, you interconnect them so there's a way for species to travel.
Yeah, so greater connectivity would help them travel between them and connect them so that we could have, you know, greater biodiversity. And in forms of fragmentation, I think in your example you mentioned, you know, 4 small patches is fragmented rather than having one large continuous patch might be better for biodiversity. Then we have connected smaller areas equals better than unconnected reserves. Yeah, so if there's no connectivity, but you have all these isolated patches and the animals from inside can't really get to the other patches, that's going to lead to lower biodiversity across all the patches. Close to other resources or other reserves. Yeah, so for this one, yeah, having high levels of connectivity, size of the patches, related to resources. So yeah, so I would go back if you have questions or about that quiz. This one's pretty straightforward, but does anybody have any questions or comments about it? Okay, if not, we can move on to number 10. So this one says distinguish between a fundamental niche and a realized niche. So why don't we first maybe start, what is a fundamental niche? How do we define that?
It's a range of conditions in which a species can persist without interference or like competition from other species.
Yeah, so we'd probably focus on the abiotic conditions specifically then, right? Okay. So the range of abiotic conditions under which species can persist, but not necessarily where they're found, right? What's a realized niche then?
It's the actual geographic range of a species where they're actually living.
Yeah, and associated with their abiotic conditions too, right? Yeah, so why does a fundamental niche, why is it not filled? Why might not we find species all throughout their fundamental niche?
Predators or competition.
Yeah, so some biotic factors, they could have superior competitors that exclude them from certain areas. And if you remove that competitor, the inferior competitor might be able to then persist in that area. Or like the other one would be predators excluding them from a certain area. They're just too easy a prey to live in the open. So they can't persist there. They got to live in more covered areas potentially. What about some abiotic factors? What are some abiotic factors that could maybe prevent an organism from filling its fundamental niche? Well, what about climate? If we want to maybe expand on that. Well, it wouldn't be sunlight or oxygen because then it wouldn't be their fundamental niche because that's the range of abiotic conditions.
Maybe their food is only accessible in parts of their livable range within certain parts of the year.
Sure, yeah, so maybe they could physically survive in a certain region, but there's no food for them.
Right, so they're
can't fill that.
traveling.
But what about like structurally? Can they always get to every piece of their fundamental niche?
Could be separated by like a canyon or a water source or some.
Yeah, that's what I was trying to get at, is it could be something like maybe a mountain that they can't cross, or they can't cross the oceans to get to a different continent. But if you planted them on that continent, they would survive and thrive. So I want to go back to the chat because there are a few questions that came in pretty quick. So abiotic factors are like soil, water, sunlight, or air. Yeah, so these are the abiotic would be non-living, so temperature, pH, salinity, those types of factors are your abiotic factors. And then I think there was, so someone said the difference kind of confused them, which is understandable, and that's why we're talking about it. So I think there was a different answer I was looking for. Yeah, so somebody says, so the difference between them is that a fundamental niche is more theoretically where they can be, and a realized niche is where the species actually is. Yeah, I think that's, in simple terms, that's a really good definition. So fundamental, the range of conditions that they could potentially survive, but not necessarily where they live. So it's more, it is theoretical. And realized, kind of in the name, they realized they fulfilled it. So realized niche is where they're actually found. And they could live, and we could say, like, you know, like a good example is 2 different continents. You know, they might be able to live in Australia coming from Africa, but if they can't cross the ocean, there's no land to get to Australia from that path. They just can't physically get there unless they're transplanted. So then they can't fulfill their entire fundamental niche. Okay, is there any other questions about fundamental versus realized niche? All right, if not, we can move to number 11. So this says, use the concept of a fundamental niche to explain why larger areas and more heterogeneous areas hold more species. So we've talked about larger areas already. So what are some, why are larger areas, you know, potentially allowed to expand a fundamental niche and hold more species? Yeah, more resources, right? Exactly. Less competition, because it could be potentially more spread out, or just having more resources reduces competition. And what is this? What do you think this means when it's talking about a more heterogeneous area in terms of habitat? So differences in habitat. So like different types of habitat, right? If you have a larger area, you're more likely to have multiple types of habitat in that given area, and having more types of habitat fulfill more species needs of different species, and therefore it can increase and hold more species. So you could potentially increase the fundamental niche if you have a larger area because it has more resources. If it's a more heterogeneous area too, you could increase the organism's fundamental niche too. Sure, I'm gonna post this in the chat real quick. Yeah, and that's a good point in the chat too. Thank you. These are the ones from Cogbook, so they're available in the modules page too. Okay, with that, unless anybody has anything to add, I think we can move on to number 12. That one says, relate abiotic conditions of a region to the species richness of that region. So again, why don't we start with simplify this and list off some abiotic conditions. What do we measure when we talk about abiotic conditions?
They're the like non-living factors like pH, nitrogen concentration, sunlight.
Yeah, precipitation is another one they put in there. Yeah, it could be rainfall, food availability, salinity, oxygen, shelter availability. So just all the non-living factors of an environment that can be measured. So now we know what abiotic conditions are. How do we relate these for a region to this? How do they affect the species richness of that region? So one answer is better living conditions, including the abiotic factors, would allow for more species. So it would increase the species richness. What do we mean by better living conditions? It's definitely on track, but make sure we all understand what's meant by it.
Like sunlight would allow for more plants versus a dark area, which would not. It would allow for more plants in that area. So it would be dependent on what we are talking about. Oh, like
Yep.
someone just said, but
Yep, no, this is great. So maybe a way to say it is more favorable or numerous abiotic conditions, the more species that that area could accommodate. So the chat we have, for example, an island with a temperature that suits multiple species rather than, for example, an extremely cold temperature that only suits very few species. Yeah, again, yep. So if it's a more commonly shared optimal temperature, then you can have more species, right? It's more common. Yeah, it could increase the species richness of that region.
So could we think of it as like the tropics, where like, you know, closer to the equator, we have a more stable climate, like less extremes versus like the poles?
Yeah, that's another way, the opposite. Think about it, it's away from the extremes, because the extremes are going to have only a few animals or organisms that can survive, where if it's more favorable or more commonly shared abiotic factors for organisms to live, such as the tropics, you're going to have greater species richness. It's a great way to think about it. Is there anything else anybody wants to add or question? All right, okay, move on to number 13. So this says, define an ecological envelope and explain the application of this concept to ecological niche modeling. Why don't we just start with the first part? What is an ecological envelope?
An area suitable for a species, but not necessarily where the species is.
How does that differ from a species like fundamental niche then? Or why would we call it an ecological envelope?
Because it's what we graph, areas that we graph that a species could be or could do really well in.
Yeah, I think you're definitely on track, and somebody in the chat got where, oh, go ahead.
Is it more of like a prediction of where a species could live?
Yeah, exactly. And that's what
Okay.
the chat said too. It's a prediction of where species could potentially live. So a realized niche is something that is actually observed, right? And this ecological envelope is kind of like a computer simulation, if you want to call it. You measure, say you take the animal into the lab and you measure what his preferred temperature is, you put it on a gradient and you see what temperature it seeks out. You put it in a non-choice test where you heat it up and cool it down and see where it loses locomotion. So those are the critical temperatures. And then you can use those type of data to put into your ecological envelope model. And then you can measure the environment as well, put out some temperature probes around and, you know, say based on the animal's performance in the lab, I would predict it to be able to live in these regions. And you can make these ecological envelopes more complicated. So I just gave an example of considering temperature, but you can also consider humidity, you can consider sunlight. If it's aquatic, you could consider things like pH and soil, or you could also put an oxygen in there. And you can even rank these parameters based on how severe they are, right? So if there's zero oxygen, but the temperature is okay, the animal can't live there, right? The oxygen is going to have a higher rank potentially than temperature because that's fundamental to it surviving.
Yes.
Yeah, so the last question in the chat was, so ecological envelope and fundamental niche are fairly similar, and I would say yes, but one is observed and one is a prediction. Yeah, so another way to think of it is an envelope is kind of like your hypothesis, and the model is the experiment. The model would be derived from experiments in the lab. So like you could, like I said, you could measure what pH this fish could survive at, you know, expose it to different levels of pH until you get a certain die-off. And then you know the range that it has to be in, and you would predict that range as part of your ecological envelope. And that we kind of already got into the second part, because that's, you know, the application of this to ecological niche modeling, right? So modeling is your computer simulation or your mathematical equation, and the ecological envelope is the output. As somebody mentioned, you could create a map, and your ecological envelope would have points where you would expect it in this map to be able to exist based on the abiotic conditions or whatever parameters you put into your model. Why do we do ecological niche modeling? Why is it a common practice in ecology? Why do we care? So for conservation
Is it
efforts,
because-
I would agree, but
is it because
go ahead.
if like a species is about to go extinct, we have areas that we know that they could thrive very well in, and we could I don't know if we have them somewhere where we're trying to build back a population, we can kind of mimic those areas so they do better at rebuilding.
Yeah, and I think that relates to the person in the chat that said for conservation efforts, right? If we want to know maybe what areas the species can thrive in, we might want to protect those areas. And if this comes to like, can we cut down this forest here or this forest there? We might say, no, that's, you know, the only area that this species can thrive in. So if we cut that down, we're going to cause that species to potentially go extinct and take away its only suitable habitat. So someone in the chat said, because it can be used to predict the expansion of certain species, like for example, pest species introduced to a continent where they haven't previously lived. Yeah, and that's the other side, right? Pest species is another big concern. So if they did a lot of this when the python was introduced into the Florida Everglades, they wanted to know, okay, this is where it currently is, it got out here, but where is it gonna go? You know, where can we predict its expansion? How severe of an impact is it gonna have? If it can't really survive the area, they might predict it to go extinct, and that's a good thing considering it invasive. But in the case of the pythons in the Everglades, they predicted to go very, you know, it's moving further and further north as we have, you know, increased temperature rises and rainfall. So it's improving its habitat, and they can predict, you know, and then based on some like of its reproductive biology, they could also add into the ecological niche modeling of how long are these reproductive cycles, how many snakes are gonna be produced each season, how far they can migrate in a season, so they can actually ask how fast can they fill its fundamental niche. Yeah, so this, it can predict the expansion of pest species, what we're just talking about, and it can predict what habitats that we want to preserve for species that are struggling or likely are facing extinction. And it can also be related to climate change, right? So as the climate is, you know, changing over history, we can make predictions into the future and say this is where it currently its fundamental niche is and where its realized niche is. But if it increases by 5 degrees in the next 10 years, for example, you know, it could expand or its range will decrease. So it can make future projections on species distributions based on all the different abiotic factors that you want to put into the ecological envelope of the model. Are there any other questions or comments about ecological envelope and how it relates to the ecological niche modeling? All right, if not, we can move on to 14. So this is the last one, and this is kind of an open-ended one. So if anybody wants to, you know, volunteer an example, so it just says, design and interpret an experiment that defines the geographic limits of a species. So if you're interested in defining an organism's geographic limits, what kind of experiments would you do? What do you need to know about the organism? Can you do it all in the field? Would some of it be in the laboratory? what are some abiotic factors that we talked about that limit a geographic distribution of an organism? So we got to answer it. It'd be a mix of both lab and field. I would agree. Like the monkey flower. So if we want to build off of that, what did they do in the monkey flower experiment? Does anybody remember that? There are a couple of videos on that. Yeah, so right, they looked at 2 different species of flower that they found growing at different elevations on a mountainside, right? And then it was basically a controlled transplant experiment where they took some species from low elevation to high and high elevation to low. And then for their controls, they dug them up, moved them around to mimic the transportation, and then replanted them. And then what did they measure at the end of that experiment? So the answer, if they could survive, right? So they looked at their reproduction in the following year, survival rate, exactly. And if they didn't survive, what would that tell us? It would tell us that maybe they potentially couldn't live in that geographic region. And if they did survive, then they knew that they could, so that maybe there was some biotic factor preventing them from getting up the mountain, maybe a competition with the other plant, or dispersal methods. So this one just got to be a little creative. I agree that you would likely measure some things in the lab, like the example I gave before. If you want to consider something like temperature, that's something for the organism that you're probably going to have to measure in a controlled setting to see what its upper and lower temperature limits are, as well as what temperature it prefers. You can do all of that in the laboratory. What are some things that you would measure out in the field then? Sure, quality of air. So yeah, you could measure the actual abiotic conditions of the field itself. Elevation, precipitation, soil content. Yeah, and these are all going to be whatever abiotic conditions you think are limiting are the ones that you should measure out in the field. So if we're talking about deer on the land, we're not going to measure salinity of the ocean next to the land, right? Because we know deer don't go in that land or in the ocean. It's not part of their, I mean, they can swim, but it's not where they're going to thrive and actually like live very frequently. So you got to think about which abiotic conditions are important to your organism of interest. And then to link this back to what we were talking about just before, you could use something like an ecological envelope to do the prediction for you through your modeling approach. So you measure, so to kind of summarize, we could measure some responses of the animal or organism in the laboratory. We measure some abiotic physical features of the landscape itself that we think it's going to potentially live at. And then we can use some type of niche modeling, such as an ecological envelope, to make our prediction of where that organism will likely be able to persist, or what geographic limits that species has. Okay, and then a question from the chat. So this is about the exam itself. So it's asking, are questions on the exam going to be multiple choice or short answer? The answer is it's going to be both. So to reiterate, the first half of the exam, so the first 50 points is going to be a mix of true, false, and multiple choice questions. And the second half, so the second 50 points of the exam, is going to be a series of short answer questions that'll range from about 5 to maybe 15 points apiece. So you'll have about 4 to 7 short answer questions on every exam. And all the exams are going to be that exact format. The first half is kind of, multiple choice and true false, and the second half is the short answer. All right, and that takes care of all of the these questions. Does anybody have any further questions? And these could be, you know, about the course itself, about the exam, about content from Cogbooks if we want to review something.
Could you give us kind of an example of a short answer question and how we should answer it?
Yeah, I think I could do that. So sometimes a short answer question might be broken into multiple parts. It might be, there might be, so it's either going to be one of two formats. It could be just straight text where you give an example or you give your answer in a series of sentences, or it could involve a calculation. You might get a table with a data set asked to calculate, say, species abundance for two different communities, and then asked a question about that based on, like, interpret your results.
Okay, thank you.
And some of them will end with explain your reasoning or why, because they want you to go in a little bit of detail of why you got your answer, what it means. And they might give you some concepts where it says define these concepts, and then it'll ask you a question of how maybe they can be related or how they can be different. So compare and contrast, as well as kind of define what they are. So they're going to be, some of them are going to be multiple part questions. The way I would kind of maybe approach the short answer questions is, if they are multiple parts, maybe break it down into each individual part and answer it separately, and then make sure to reread the question at the end to make sure you answer each of the parts. And you're allowed to use scratch paper on the exam. So if you want to, you know, just write out some ideas and, you know, write different parts of it before putting it on the computer, you know, that's maybe one strategy to do too, so you don't accidentally submit it. But
Now, for the short answers, and they could be a range of points per question. Is it like an all or nothing point scale, or is it you can get part of the answer correct and part of the points? Or
yeah, I definitely will try to look for giving partial credit. So you can get, it's not all or nothing. You can get partial credit.
okay, awesome.
And if you get, say you get 9 out of 12 points, myself or the person grading it, because the TAs are going to be grading the short answer questions too, they should explain why you lost the points. They might say, like, you defined this term correctly, but this one wrong and this one correctly, so you got, you know, 6 out of 9 points.
Awesome, thank you.
So the question was, can we view all the questions at once, or do we only see them one at a time? You will only see them one at a time, but you can go through back to questions or skip questions. And the other question was, will you do other sessions going over content throughout Cogbooks, or will you only do exam prep hours? What we do in these office hours is essentially up to the class. So, you know, I don't have anything planned for these office hours. I like to leave them open. A lot of times students do like to go over the questions from Cogbooks. Sometimes in future activities, you're going to have more involved activities in the Cogbooks, and sometimes students like to go over the activities or ask why the answer is what it is, and we can discuss that as a group. So they kind of, again, like I said, that they'll be up to what the students want to do during these office hours. The other question was, so the questions in Cogbooks are pretty similar to the exam questions. I'd say it's probably a mix. Some of the questions are going to be almost identical with different numbers, asking, especially when it comes to things like the calculations. Some of them are going to be more conceptual, so you will have to be able to put pieces together, given part A and part B, asked to connect them through part C. So just kind of think beyond defining terms, think about comparing and contrasting terms, relating them or explaining them, or maybe like the question 14 was kind of open-ended, right? That says design an experiment to answer this type of question. So that is going to be very more conceptual and open-ended on your part to fill in. Are there any other questions? Okay, if not, then I'll go ahead and we can call it for this office hours, and then I will be posting this video shortly. It takes a little time to download and upload to the system, so truthfully, it'll probably take close to an hour before it's up. And no problem. I thank everybody for coming and thank all of you for participating. I'm sure your classmates that couldn't make it are going to be appreciative of this. So remember the exams tomorrow, and let me know if there's any technical issues. I just recommend don't wait until the last 3 hours that it's open, because sometimes there are some technology issues. So with that, good luck on the exam, and everybody have a good night.